Everybody, in this video we are going to take another look at VDJ recombination. This is going to be part 2, and a phenomenon called allelic exclusion, which is related to VDJ recombination. Now, before watching this video, make sure you watch videos from the previous lecture where I covered bits of a DNA repair process called non-homologous end joining and the related process called microhomology mediated end joining. And of course, VDJ recombination part 1. So you're going to want to watch those videos and make sure you understand this stuff so you can understand these things, which I'm going to talk about in this video, where these could be rather complicated. So I encourage you to make sure you understand this stuff first before you try and understand this stuff. Now, just a quick, rehash of what we went over in VDJ recombination part 1, the previous video. So in mammals like humans and mice, we have 3 different loci in our genomes that encode the light chains and the heavy chains of the immunoglobulins, so antibodies that our B cells produce. And we spent a lot of time talking about these loci in the previous video. So we have the IgK loci, or locus here. We have 2, right? We're diploid organisms. So we have the IgK locus and the IgL locus, and these are in charge of making or encoding the light chains for antibodies. And the IgH locus, now this is in charge of encoding the heavy chain for antibodies. Now, these aren't really traditional genes, right? So these are made up of gene segments, and the gene segments need to be rearranged or recombined to make a functional gene. So with VDJ recombination, what we have is one of the V segments, and there could be many of these V segments. I've only diagrammed 3 in each case, are linked to one of the J segments. Now, IgK and IgL are different from IgH in that they don't have the D segments down here. So IgK and IgL, it's a single recombination event involving joining a V to a J. Same thing for L, one of the V's to one of the J's. Now the IgH is a 2-step recombination process of joining one of the D's to the J, a J first, and then one of the V's to that same D segment that was joined to the J. So this would be step 1 over here, this would be step 2 over there. And we went over this in pretty good detail in the previous video. But we didn't really cover the mechanism of how it happens, how the cell actually breaks the bonds and links them together. And that's what we're going to cover in this video. So now the mechanism relies on a number of different proteins, but the most important ones to get started are called RAG1 and RAG2. Now these are recombinases, so the RAG recombinases. These form a complex, the RAG1, RAG2 complex, which is, initiates this whole process. And another key thing we will need to learn is these RSS sequences. So this stands for recombination signal sequences. Recombination signal sequences. Okay, now the RAG1 and RAG2 recognize RSSs, and they recombine the V and D and J segments according to a rule called the 12-23 rule. And this will make more sense in a moment. So let's take a look at just the IgK locus. Now, I'm going to quickly diagram 3 variable gene segments. And sometimes when we're talking about V segments that are specific to the K locus, I can put a little K down here next to that V. And, there can be many more of these, and we alluded to, how many of these there can be in human or mouse, I think, and getting the exact number is sometimes difficult, but I think I just said more than, let's say dozens of these. But I'm only diagramming 3. Now the J segments down here, I'm also only going to diagram 3 over there. And then we have the constant segment down here. Now there are or segments. I think of the K locus, there's technically only one constant segment. Now the constant segments, these, we're going to learn more about these in the next lecture. These are involved in class switch recombination, which is different from VDJ recombination. So now we have some V segments up here from the IgK locus, and we have some J segments down here. Now each of these V segments at the IgK locus is going to have an RSS sequence. Downstream of the segment. And so in humans, each of the VK segments has what we're going to call the 12 RSS. Now each of these J segments, I didn't want to put those lines in there because it's going to mess up my triangles. These triangles represent the RSS sequence. So the J segments, each of them have upstream, immediately upstream of the segment, a 23 RSS. So the RSSs come in 2 types, 12s and 23s. At the IgK locus, each VK segment is followed by a 12 RSS, and each JK segment is preceded by a 23 RSS. Now, the IgL locus is very similar. But interestingly, the VL segments are followed, each VL segment is followed by a 23 RSS, and each JL segment is preceded by a 12 RSS. Now, IGH is going to be a little different than those two because we have 2 recombination events. So the organization of the RSS sequences is going to be a little bit more complicated. So let's just take 3 variable segments from the H locus and some diversity segments. And it's very common not to put this little h in here when we're talking about the IgH locus, because the IgH locus is the only one with diversity or D gene segments. So we can leave that H out. And down here will be some J segments. Now at the IgH locus, each of the VH segments is going to be followed. I'm going to put the RSS sequences, I'm going to indicate those with a little arrow here, or a triangle. Each of these is going to be a 23 RSS. 23 RSS, 23 RSS. So every one of these VH segments at the IgH locus is going to be followed by a 23 RSS. Similarly, all of the JH segments at the IgH locus will be preceded by a 23 RSS. So each of these has a 23 RSS. The diversity segments are preceded and followed by 12 RSSs. Okay, same thing, 12 RSS here before and after each of the D segments. Now the RAG recombinases, the RAG1 and RAG2 complexes, or the RAG1 and RAG2 complex, they will choose might be easier with IGL or IGK because we don't have to worry about diversities. So what the RAG1, RAG2 protein complex will do is it will bind, one of these segments at the RSS sequence, and it will bind one of the JK segments at the 23 RSS segments, and it will rearrange those. So it will fuse one of these segments to one of these based on binding to the RSS sequences. And it can only fuse a 12 RSS to a 23 RSS. More accurately, it can only fuse a sequence linked to a 12 RSS to one that is linked to a 23 RSS. That's why it can't recombine two VK segments or two JK segments, because each VK has a 12 RSS and each JK has a 23 RSS. That's the 12-23 rule. So the RAG1, RAG2 recombinases, they fuse one sequence that's linked to a 12 RSS to another sequence that's linked to a 23 RSS. So that's the requirement of this recombination process. Now, when it comes to the IGH locus, what happens is the RAG1, RAG2 recombinases use the 12 RSS from one of the D segments to fuse D to one of the J segments with a 23 RSS. Then after that's done, it can use the other 12 RSS on the D segment to fuse that D segment to one of the 23 RSS gene segments, one of the VH gene segments that's followed by a 23 RSS. So we're going to look specifically at what these 23 RSS and 12 RSS sequences look like, so you can understand. So if you're a little confused now, I think it'll become clear in the in a few moments. So now, okay, so here, let's assume we're looking at the IGK locus. And here we're looking at just one of the V segments. And here is one of the J segments. And if you remember, when these are rearranged, when these are recombined, it's just fusing one of the VKs to one of the JKs. And I didn't diagram any of the other variable segments or any of the other JKs. We're just going to assume the cell's going to pick this one, it's going to pick this one, and it's going to fuse these together. These purples things here, these are the recombination sequences here. So this one's going to be the 12 RSS, and this one over here is going to be the 23 RSS. Do I have that right? Yes, I believe I do. Now, I want to show you how the cell fuses this gene segment to this one over here. Now remember, there are others in here that we're just ignoring. So we're assuming the cell picks this one, the RAG1 and RAG2 recombinases, randomly chose this one to fuse to this one over here. Now, so I'm going to show you some details on what the 12 RSS sequence looks like. So the 12 RSS sequence is in purple here. So the 12 RS sequence is going to be in general, the RSS sequences have a heptamer sequence, so a sequence of 7 bases, followed by the 12 or 23 base pair spacer, followed by a nonomer sequence, so 9 base pairs. So this is the consensus sequence of the heptamer. So most RSSs have a CAC, AG, TGC with the 5' end up here and the 3' end down here. So that's CAC, AG, TGC in the complement down here. Now that's the heptamer. So the nonomer, most RSSs have this sequence for the nonomer. ACA, AAA, ACC. So there should be 9 here. Let me see. 1, 2, 3, 4, 5, 6, 7, 8, 9. Yes, 9 nucleotides here and 9 base pairs. So here's the complement down here. So those are the consensus sequences, and most RSSs have this sequence for the heptamer and this sequence over here for the nonomer. Now, this is a 12 RSS, and so the 12 is the spacer, like how many base pairs are found between the heptamer and the nonomer. In this case, it's 12 because this is a 12 RSS, and these are highly variable. So for our purposes, it doesn't really matter what the bases are in here as long as there are 12 of them. So heptamer of this sequence, nonomer of this sequence, 12 base pairs, this will be recognized by the RAG1, RAG2 protein complex. It will bind to this sequence. Now, what we have here in red, so I just picked, 5 base pairs here. And what I'm saying is this is part of the coding region of the VK gene segment. So the RSS heptamer, that's not part of the coding region. That's going to be removed. So this is part of the coding region of the VK segment. This is where the RSS starts. This purple region right here, this is the RSS. That is going to be separated from the coding region of the VK gene segment. And we can follow this all the way. Now remember, this VK, this could be very far. There could be lots of other VK segments in here. There could be lots of K segments in here. And I didn't diagram them, but what you can see down here is I put this little, I put some random bases here, some random bases here. I put this dash here, meaning that who knows what's in there. There's a lot of stuff in there that I'm not diagramming and not detailing, because what we want to focus on is we want to focus on this RSS over here. So this is the 23 RSS of the JK segment. Now, the consensus sequence is identical, but it's in an inverted orientation, right? So we have CACAGTGC here. Now look down here. This is the 5' end, right? CACAGTG. Sorry for the messy writing in there. So it's just inverted relative to this guy over here. This is going to be the heptamer. This is going to be the spacer, which is 23 base pairs in this case. And this is going to be the nonomer sequence. So it's just inverted relative to this one. Now, you can see in yellow here, I just picked some nucleotides of the JK segment here. So I just picked the first 5 nucleotides, CCGAA. They might be CCGAA, they might be something else, it doesn't really matter. The important part is that this RSS sequence will be separated during the recombination process. It will be separated from the coding portion of JK during the recombination process. Now, again, the consensus sequences of the heptamer and nonomer are consensus sequences, so they're nearly identical, we should say, or highly similar between all RSSs. Doesn't matter if you're 12 RSS or 23 RSS. However, the spacers, it doesn't matter what the sequence is here. As long as it's 23 base pairs long, this will function as a 23 RSS. So the RAG1 and RAG2 recombinases bind to these RSS sequences. And they're able to fuse this one to this one because this is a 12 RSS and this is a 23 RSS. And the process by which the RAG1 and RAG2 complex chooses segments should be random. And that allows for the diversity of light and heavy chain sequences as this process is random. And we're going to see in a moment of how this process leads to even more diversity than just joining random fragments together. Okay, so after the RAG1 and RAG2 recombinases bind the RSS sequences, what they do and I have heptamer and nonamer labeled here for us. What they do is they nick the DNA between the heptamer and the coding sequence. So we're going to nick right here between this G and C right here. And it's going to nick between this G and C right here. And it's also going to do some other things, because this nick isn't just going to remain a nick inside the nick on the DNA strands here. What it's going to do is it's going to use that nick and it's going to fuse it in a way that makes this end, this coding strand right here, this G and this C, after the RAG1, RAG2 nicks the phosphodiester bond between this G and this C, between the end of the V segment and the beginning of the RSS, is going to link this G nucleotide right here to this C nucleotide right here. This is going to be called the hairpin coding end. And, you know, and I don't know for sure, but I doubt that there's actually hydrogen bonding going on now between this G and this C. But the important thing to know is that all these nucleotides are intact, but this G is now linked to this C, and this is a hairpin now. So the phosphodiester backbone goes this way and then circles around and goes that way. So this is a hairpin coding end. However, the other end, this is the end of the RSS now. So see this CACAG, CACAG. It's just a loose DNA end here. This is called a signal end. So at each side right here, we have a hairpin coding end and just a loose, just a double-stranded end right here. Just a free double-stranded end with a phosphate group and an OH group right here. This is called the signal end. So we got a hairpin coding end and a signal end. Same thing happens on the other side. So we get the break here, and then this TTCGG, TTCGG. Oh, I didn't color that in. So this phosphodiester backbone right here is now going around without any breaks. There's no loose phosphate or hydroxyl here. This is a hairpin coding end. And same thing for the RSS sequence. Well, not same thing to this one over here. The signal end is just a blunt end with a free hydroxyl group on the 3' end side, nucleotide right here, and the 5' nucleotide has a phosphate. Now, I have the RSS's colored purple. The non-colored ones, I don't know if this really shows up in the video, but there's no color down here. So this is all that intergenic sequence that is lost when a VDJ recombination event happens. So maybe you remember, if we were to fuse this one right here to this one right here, all this stuff is lost. So all that sequence is down here, and these are connected, but they're going off of the page here. So after this has been processed to give us 2 hairpin coding ends and 2 signal ends, well, then non-homologous end joining takes over. So RAG1 and RAG2, those guys, essentially for our purposes, we consider those guys, they've done their job. Now, I've got a lot of this stuff diagrammed out for us already. So let's take a look at what happens to the signal ends. Well, scientists aren't exactly sure what happens, but what they think happens is those 2 signal ends are grabbed by the non-homologous end joining proteins, like the Ku70 and Ku80. We didn't talk about too many of the proteins involved in this process, but it's thought that they will just grab these ends and they will link them together. ligate them together. So one of the ligases involved in non-homologous end joining will ligate those together and make a circular DNA molecule. Again, all of that stuff that's lost, all of that DNA that's lost, that's between the V and the J segments that are being joined, all that's going to be down here, part of this circular molecule that is just going to be degraded by the cell at some point. So the exact degradation process is not known, but what is known is that this is lost. It's no longer part of, if we're talking about the IgK locus, it's no longer part of the IgK locus. Now, the important part that we need to consider is what happens to those coding ends. Now, so non-homologous end joining, the proteins involved in, joining double-stranded loose DNA ends, is going to process the hairpin coding ends. Now, when we talked about non-homologous end joining in the previous video, we were talking essentially about a double-strand break in a DNA molecule. We didn't talk about, hairpin ends. This is something rather kind of unique to VDJ recombination. So the non-homologous end joining proteins have to do something kind of, unusual. So the first thing they do, and so here's the diagram of, and I can show you how I'm just continuing on from the previous diagram. So here we have the V segment. Now here I diagrammed the V segment kind of small, but it's actually quite long, and I'm just diagramming a few nucleotides at the end. Now see, we have the TACGG over here, and for the J segment we have CCGAA, CCGAA. Okay, so these are the same sequences. I'm just using the same example here, and I'm zooming in, and we can see that we have the hairpin end here. The G is linked to the C through a phosphodiester bond. And this G down here is linked to this C through a phosphodiester bond. And they don't have to be GCs. Just by random chance, I chose those nucleotides. So the first thing the non-homologous end joining proteins have to do is they have to eliminate the hairpin. And so they do that just by causing a nick, putting a nick anywhere around here. They got to nick it and change these hairpin ends to just regular DNA ends. And in this example, I'm choosing right here. Now, so this process is random, and that's going to lead to some of the diversity we're going to see that can result in the after recombination. So I'm going to assume non-homologous end joining chooses right here between this A and this C to break the phosphodiester bond. And for over here, I'm going to choose, where did I choose? I'm going to choose right here. So it has to choose somewhere close to the end, it has to nick the phosphodiester backbone of each coding end, each hairpin coding end. And when it does that, it causes the bases to flip out. So if we break the phosphodiester backbone here, this GGC is going to flip out, and now we're going to have this. Okay, see, ATGCC, GGC, ATGCC, GGC. Okay, can everyone see that? Same thing for this one here. If the phosphodiester backbone is broken here, these are going to flip out and give us like this overhang here, right? So now we have TCGGCCGAA, TCGGCCGAA. Okay. Now, when these flip out, what we're going to call these new bases that were now, they're sort of extensions of the bases that used to be here. These are called P nucleotides. So we have P nucleotides on each end here. And the P is for palindrome. Palindrome. Or palindromic nucleotides. So now once we have the strands nicked and we have our P nucleotides, a lot of things can happen. So now, when we talked about this a little bit when we talked about non-homologous end joining, so essentially these ends need to be processed so that they are blunt. And we can achieve non-homologous end joining can achieve that in many different ways. So this could be filled in, like I've shown all the way down here. Or all of these bases can be removed. They can be trimmed away. Either way, we can trim away some of them and leave some behind. Non-homologous end joining can do many different things. I'm just showing one example. In the example I'm just showing that non-homologous end joining is filling in the ends. So we're going to keep all these nucleotides down here. So GGC is the P nucleotides. They're now base paired with CCG right here. Okay, so now we have blunt ends on each side. Non-homologous end joining proteins filled this in here. One of the polymerases involved filled this in here. And so now we have two blunt ends. And we could even put a 3' OH here, a 5' phosphate group there. And now these are ready to be ligated by DNA ligase. However, another thing can happen. So another enzyme can come in, and that enzyme is called TDT, for terminal deoxynucleotidal transferase. And what this does, this adds nucleotides. And we have a range of nucleotides. In the notes, I say 2 to 5. I don't know where that reference came from, because why can't it add 1? That doesn't make sense that where that 2 to 5 came from. So I'm going to say 1 to 5 nucleotides. The TDT enzyme can add to the ends of the processed coding ends. So if we're continuing from this step, where non-homologous end joining had made the ends blunt, so we have our CCG on this side and TCGG on this side, CCG on this side, TCGG here. Well, TDT, it can add nucleotides at random to the 3' ends. And in this case, I put 5 on each side. So I just adds them randomly. It could be any combination of nucleotides here. And it may add some, it may add none. So completely random process. And okay, so let's say it does add these. Well, now these have to be joined, right? And so these could be trimmed away, and then it could be joined in any number of ways. But let's say that these are not trimmed away. And remember, we have that variation on non-homologous end joining called microhomology-mediated end joining. Well, let's assume that works here. And what happens is we can have some base pairing occurring between the bases in here and bases in here. Even though it's not perfect, we can get some base pairing occurring, and that's the microhomology aspect of that name. So we have a GA here and a CT here. So let's assume some base pairing happens between there. So we have GA here and CT here. Now, what NHEJ or MEJ, whichever one you want to assume is working at this process, all it needs to do now is fill in these gaps and use a ligase to seal the nicks. So, and I have that written here, fill the gaps. So, okay, so what happened after all that? Okay, so now we have, I'm going to show you what happens, let's say, remember these were the original hairpin ends. Now, imagine the cell could just join these together without any P nucleotides or N nucleotides. Well, it would look like this. But as we saw, we can end up with something that's very different from here. So what we have is, this sequence over here, this sequence over here, because of the process of adding the P nucleotides and the N nucleotides, which is a highly random process, we end up with a very different sequence than what we would have what we would have ended up with if those segments were just joined together without any additional processing. Now, these extra nucleotides, these are P nucleotides here, these are the P nucleotides, these are the N nucleotides. All this leads to, all these P and N nucleotides, it leads to increased junctional diversity. So increase in the number of sequences that can exist between the V and J segment junctions. And so because we're looking at V linked to J, we're talking about light chains. So here I'm showing you, okay, this is the variable region of the light chains. And when in one case we would have just red and yellow, but now we have all those blue and green and blue sequences in here as a result of the particular mechanism used to process these ends. So let me say that another way. So let's say RAG1 and RAG2 chose V segment number 25. You know, let's say there's several dozen of these or 100 V segments to choose from, and it focused in on V number 25, and it focused in on J number 7. It fused this one to this one. Here are the RSSs. Now, without the P and N nucleotides, every time it settled on those, we would get the same sequence every time. When there are lots of different V segments and J segments, so there's still many different light chains we could get from joining those together, many different sequences we could get, but it's somewhat limited, right? Somewhat limited by the total number of V's and total number of J's. However, when we add the P and N nucleotides in there, I mean, I don't know what the number is. I'm just going to say a huge number of possible sequences can result from joining V number 25 to J number 7. We're not just limited to this. The cell has a huge number of sequences that can result from trying to fuse V number 25 to J number 7. And the same thing happens with every other combination. So this increased junctional diversity is essentially the reason why, is one of the reasons why our B cells can make antibodies that can essentially recognize any antigen, or learn or adapt to recognize any antigen. So really, I think it's really a fascinating process that evolution has given us this mechanism to be able to recognize such a wide range of antigens. Antigens that don't even exist yet. So we're not done yet. I hope you understand how that process works. Very confusing. And I essentially detailed all of that in the notes, essentially as I just kind of diagrammed for you for this video. Now what I want to do is talk about this phenomenon called allelic exclusion. So, and what does allelic exclusion mean, and why is it necessary? So this is a really fascinating mechanism too, and scientists really don't have a good explanation for how the cells do it. But okay, so allelic exclusion. So, and it helps to, well, first to help me introduce this phenomenon, let's take a look at those V and J segments that we were already looking at in an earlier part of this video. So here's the end of the V segment, here's the end of the J segment. Now, let's assume, and this is possible, is that these could be joined together without adding any P or N nucleotides, right? Because that's essentially a random process. The amount of trimming that's done, where the hairpin coding ends are nicked, if any N nucleotides are added, all of those steps are random. So it's possible that these could just be joined together. Now, let's say this ATA is one codon, CGG is another codon, CTG is a codon, and AAA is a codon. So these code for specific amino acids of the light chain. And we have I followed by R, followed by L, followed by K. Now, because of the way these ends are processed by adding P nucleotides and adding N nucleotides, often adding P nucleotides and or N nucleotides, we can end up with something like this. So we have the V segment end, the J segment end, and what I'm showing you here is 2 P nucleotides that might happen to be inserted, and 1 N nucleotide that might happen to be inserted. In this case, well, we have a codon here for I, codon here for R, and this CCA happens to be a P. The P isn't for P nucleotides, it's for proline. So this codes for an extra amino acid. And we have L here and K here. Okay, so this is a light chain. Seems like this recombination with these extra P nucleotides and N nucleotides would lead to a light chain with an extra P than what you would normally expect just by joining those fragments together. And okay, that makes sense. And you know, maybe that would be a good light chain for recognizing a particular antibody, antigen. Sorry. However, because adding in P and N nucleotides is random, we can get anything can happen between the V and J junction, right? So in this case, I'm showing what would have happened if we added just 2 P nucleotides without an N nucleotide. So we still have one codon here, one codon here. And you know, how am I getting this? So this is I'm using single amino acid codes for the amino acids coded for by these codons. And you can look up ATA in a table, and CGG in a codon table, and CCC in a codon table. And I forgot to introduce that when I was going over the previous diagram. And this would be the top strand would be the coding strand. So, okay, so where am I going with this? Okay, so here we have a codon, it's okay, this one's intact, this proline, okay, that looks okay, but look what happened. Because we only added 2, we ended up shifting the reading frame, and now the next codon after this CCC is a TGA. So this is a stop codon. So this recombination, you know, even though we sailed those hairpin coding ends together and added 2 P nucleotides, if the cell, you know, tries to transcribe this into a messenger RNA and make a light chain out of there, it's not going to work. It would not encode a functional light chain because it has this early stop codon. We're missing like this whole J segment, and now we're going to be missing the constant region also. So this is called a non-productive recombination right here. And it's useless. And the cell has a way to determine when a recombination event is non-productive, when a VDJ recombination leads to a non-productive recombination and does not produce a functional protein. Now, scientists don't really know exactly how cells determine that. Well, as far as I know, they don't really know how cells can determine that. But it leads to this phenomenon called allelic exclusion. So what is allelic exclusion? So essentially, allelic exclusion, now, as we talked about already, we have IgK loci, IgL loci, and IgH loci. These are in charge of light chains. These are in charge of heavy chains. So during B cell development, the developing B cells try to do the VDJ recombination at the IgH locus first. Now, humans are diploid, right? And the IgH locus is found on chromosome 14. So here we have, let's say, 2 chromosome 14s in a random immature B cell. So a B cell that's just starting to develop. Now, let's say this is the IgH locus. I'm just diagramming it, putting it at the top of the chromosome here. I don't really know where it is in chromosome 14. And the H0 stands for, let's say, the original. O is for original. I should say HO, for the original arrangement of the IgH locus. So the germline arrangement before any recombination has happened. So what the cell will try to do is recombine one of these. So we'll choose one at random. And if it works, let's say productive, what will happen is I'm going to change this to a plus. Let's say this H0 right here. The cell tried to recombine the IGH locus right here to make a functional heavy chain, and it worked. So I'm going to say H plus. This other one will stay in the original arrangement, the germline arrangement here. And so this one does not make a heavy chain because it never underwent recombination, so it doesn't encode a normal messenger RNA that makes any sense. This one here does. Okay, great, so the cell is done, good. It just stops right there, and this one always stays in the germline arrangement. However, if it's not productive, so if let's say the addition of the P and N nucleotides led to the insertion of a stop codon or a frame shift that led to a non-functional protein, what would happen is we're going to call this one H minus now. Somehow the cell knows that that failed, and it will try again over here. So it will have to try with this one over here. It only has one more shot at this. It's got one chance now. And if it's productive, okay, so then we have one failed rearrangement at one of the IGH loci, but one that worked. Okay, so this cell is going to be happy. It has a functional heavy chain. If it was not productive, okay, well then the cell is unhappy. And then apoptosis is initiated because this is a B cell, and a B cell that can't produce an antibody is a worthless B cell. So this, where both recombination attempts at the IGH locus failed, leads to programmed cell death. So that poor cell is not going to make any descendants. It's over. It's done. So, but the body can usually make lots of different B cells, so it will try again with a different B cell. So that's how allelic exclusion works for the IGH locus. What it means essentially is that only one allele is going to work, the other one is not going to work. So as a result, if we should look at the H chains of the antibody made by this one cell, they are going to be identical. So even though there are two IGH loci, only one of them is going to be used, only one of them is allowed to form a productive rearrangement. If one of them does, then the other one isn't even an arrangement. If the first recombination succeeds, then the other one, the second one, isn't even attempted. As a result, all of the heavy chains are identical. So the variable regions of the heavy chains are identical. These are identical proteins because there's only one IGH locus that was recombined in a functional way, in a productive way, and there's only one type of protein that can be made from that productive recombination. So these will be identical, N-terminal regions here, C-terminal regions down here. Now, allelic exclusion also works for the light chains. However, we have two different loci, right, for the light chains. We have the IGK locus and the IGL locus. IGK and or IGL recombination, so allelic exclusion there. So only one of these can be used. So, in total we have 4 different loci that could produce a light chain. Only one of them is used. The others are excluded. And how does that process work? So after the H chain, heavy chain has been produced, and the cell produces a functional heavy chain, it's going to try one of the IGK loci or one of the IGL loci. I think in humans, the cell tends to start with IGK. So I have the cell trying one of the IGK loci first. If it's productive, great. Okay, so the cell's done. None of the other loci. If there are 2 IGKs, right, because we're diploids, and 2 IGLs in that B cell, if the first one works, the others are not recombined. And somehow how the cell knows to stop recombining is really unknown. But it stops, and only the productive recombination of the IGK locus, only the single productive recombination produces a light chain. And that's why when we have our antibodies here, the H chains are identical. We already saw why those two are identical, and that's why the two light chains are identical. So heavy chains here, light chains here. Only one of the alleles is allowed to make a productive rearrangement that encodes a functional protein. So that's why B cells only make one type of antibody, even though they have 4 different loci that can make a light chain and 2 different loci that can make an H chain. They only make one type of antibody because of this allelic exclusion. Only one of the light chain loci is allowed to make a functional protein, and only one of the heavy chain loci is allowed to make a functional protein. So let's say that first attempt with IGK failed, not productive. It's going to try the other IGK. If it works, good, it's just going to stop. If that doesn't work, it's going to go on to the IGL. It's going to try one of the IGLs. If it's productive, great. If it's not productive, well, it's going to try the other IGL. So if this fails, none of the light chain loci produced or resulted in a recombination that makes a functional, that can produce a functional light chain, well then the cell has to, will undergo apoptosis, programmed cell death. Okay. So that's essentially how allelic exclusion works. Again, scientists are still trying to work out the details of how this cell, knows when to stop trying recombination, how it's able to focus in on one locus at a time. All these details are relatively obscure. And hey, if you go into research someday, you can become pretty famous if you can figure out how that process works. Okay, so I hope you understand how VDJ recombination works, how the RAG1 and RAG2 complexes work, how the P and N nucleotides lead to junctional diversity, and how, allelic exclusion works. And in the next video, we will take a look at class switch recombination, which allows the cells to use the same antibody but switch the isotype of the antibody. So recognizes the same antigen, but changes the isotype of the antibody. And that might not make much sense right now, but hopefully it makes a lot of sense when you watch the next video. We'll also talk about somatic hypermutation and how the molecular processes involved in sort of helping or improving the ability of an antibody to recognize an antigen. Okay, see you in the next video.